Thank you very much for the introduction. Good afternoon, ladies and gentlemen. It's really a privilege to be here, and also an honor to show you the results, the final results of this study. When we show and we try to find out a good acronym of a study, at times it's always difficult. This time I think that EUPHRATES tells us something, because this is the name of the river that represented one of the cradles of the civilization, with the augury of having find the possibility of, through this study, of having a multiplicator of knowledge. We'll see. So the EUPHRATES stands for Evaluating the Use of Polymyxin B Hemoperfusion in a randomized controlled trial of adults treated for endotoxemia and septic shock. The principal investigators are listed here, and I'm again honored to be part of the steering committee. When you look at the endotoxin, we are speaking about a real powerful trigger of sepsis, and whatever would be the perspective we are looking at the endotoxin as a molecule, this can be a damage-associated molecular element or a pathogen-associated molecular patterns. But in any way, during sepsis, it has the capability to link the sulfase receptor, and then through the transducers of the signal, including the NF-kappa B, translate the signal to the nuclear acids to produce several mediators that are dangerous and harmful for our organism. And there's something more, that endotoxin activity can tell us that the endotoxin is an independent marker of the ICU mortality. In other words, the higher is the level of the endotoxin, the higher is the risk of the mortality. One important point is that when we have a mediator, it's important to have a tool to measure it. And if we want to illustrate a mean to remove the endotoxin from the body in patients with septic shock, this has a fundamental issue and represent a logical therapeutical approach. The threshold for the endotoxin determination for those patients who are in septic shock is represented by a level of the assay which is about 0.6. And the principle which is behind the removal of endotoxin exploits a cartridge that through a ionic and covalent binding through the polymyxin, which is adherent to the fibers of the cartridge, can remove the endotoxin from the circulatory torrent. The priming volume is 135 milliliters. The weight of the fibers is slightly 56 grams, and the concentration of polymyxin B, which is not liberated into the circulatory stream, but is tightly adherent to the fibers, is something like 5 milligrams per grams. We have an extracorporeal circulation that passes through the thoramyxin filter, having a blood pump whose flow rate is something in between 18 and 120 milliliters per minute. The duration of the treatment is roughly 2 hours, and the dose of the treatment consists in 2 treatments over 24 hours. The thoramyxin has been applied over the years in many, many different patients, and there are several reports of prior investigations, including and treating more than 150,000 patients worldwide. And its use, it's relatively old because it started more than 20 years ago, The safety, at least looking at these reports, is quite good. Why? Because the serious adverse event rate was quite low, and has been published so far more than 150 articles reporting the use in 7,600 critically ill patients. What was the protocol design? The intent was to evaluate the safety and efficacy of the PMX cartridge in a phase 3 double-blind randomized controlled trial that compared the standard of care plus the PMX cartridge versus the standard of care. The main characteristic of this study was that had also an adaptive design with an interim analysis for the reassessment of the sample size. The protocol design was driven by the biomarker, which means the determination of the endotoxin activity that was used to select the endotoxemic patients, and all those patients who had a diagnostic test that was negative were indeed excluded. The protocol design addressed a specific population that was the one of septic shock patients having a multiple organ dysfunction score above 9 and having an endotoxin activity level above 0.6. All the different data were centralized by the coordinating center that confirmed eligibility prior to the randomization. The timeline consisted into the identification of the enrollment criteria and having the screening of the endotoxin. Then, after having the approval from the central center, the patients were randomized to receive 2 treatments, and the time interval between the 2 treatments was 24 hours. And 94% of all patients were treated following this timeline. The 2 treatments evaluated as a treatment specification, the standard of care plus the 2 polymyxin cartridge treatments, and the sham group of patients received, as said, the standard of care plus 2 sham hemoperfusion, which means a fake catheter without any extracorporeal circulation. And one of the interesting things was the design that was conceived blinding completely the investigators and the attending physician together with the steering committee, the SM, and the sponsor, and having as unblinded team the ICU nurses, the local study coordinator, and the nephrology team. This is something, for instance, that could be done in Europe, but in North America, simply because in most of the cases in Europe, the extracorporeal circulation and CRRT is run by the intensivist, and the nephrologist act in collaboration, but not the ones who had the direct responsibility. Blinding was successfully maintained in more than 97% of the cases of all subjects. The primary endpoint consisted in safety and efficacy based on the 28 days mortality, and we had a number of secondary endpoints consisting in the survival time from the baseline up to the 28 days, the improvement of the multiple organ dysfunction score from the baseline up to the day 3, the change in mean arterial pressure following the same timeline, the change in cardiovascular index, which is a cumulative vasopressor index, which consists in the combination of different vasoactive and inotropic drugs that can be given and administered to these patients. And the formula reported in the lower part of the slide illustrates how it was calculated. And there was also the consideration of the ventilator-free days and the renal replacement-free days. as well as the survival at 14 and 90 days and at 1 year follow-up. The population initially screened consisted in more than 900 patients, randomized 450, and those who had MODS above 9 were 295. Ultimately, there were 244, 115 receiving polymyxin filter, and 129 the sham belong to the sham group. If you look at the demographic, in large part, the age of these two groups was roughly 60 years. Looking at the age groups are quite evenly distributed, and they were prevalently male, Caucasian race that represent mostly the population in North America and in Europe. Looking at the various levels of the cardiovascular index, again, the distribution of the different subcategories was quite balanced between the two groups, as well as the renal replacement therapy and the presumed site of infection. Interestingly, if you look at the different site of infections, were included not only those patients affected by intra-abdominal infection, as it was in the previous study that was accomplished in Italy and published years ago in the JAMA, but also patients who had infections localized in different parenchyma, like the lung. And also, interestingly, looking at the different microorganisms, we had something like 34% of the group that had no growth at all, and some of them had a mixed growth of bacteria. And the presence of bacteremia was quite balanced, roughly 30%. The safety of the treatment was indeed quite good, and only 8% of the cartridge reported some clotting that indeed this is a very good, especially remembering that other negative trials in previous years showed very high and higher incidence of clotting. All the serious adverse events reported into the table had indeed not related to the treatment. If you look at the population randomized analyzed, we have the intention to treat population that is represented to the upper part of the table. Then the intention to treat population with patients having a multiple organ dysfunction score above 9, and the per protocol population with all patients who had a MODS above 9 and who completed the treatment. The other point that if you look at the primary endpoints, at least in the global analysis, indeed the study was negative. However, if you look to the analysis on the pre-specified, as pre-specified, all those patients who had a MODS above 9 and received 2 full treatment, there was a mortality rate at 28 days that was not different, but there was a trend in favor of the group who received the polymyxin filter. In this case, almost 32% versus almost 37%. But if this is true, I think that we have to ask some questions. There were some primary endpoints that were not met, and then we have to ask if the study was underpowered, if the effect size was overestimated, or if there were confounding factors. However, we have also to consider that this is, in fact, the first trial that is biomarker-driven, and this biomarker, as the assay is not perfect, may have some potential confounding factors. And we need also to remember that the patient population was selected on a minimum endotoxin activity value, always in septic shock. The point that is behind, at least just going deeply into further investigation on the population and other results that are collateral but very interesting, is that probably the endotoxin burden in certain patients may be too high, and we are not sure if this was measured accurately or treated effectively. Indeed, there is a sort of balance that implies the different distribution of endotoxin through the endotoxin removal, the physiological excretion, the volume distribution within the body, in the tissue, the interstitial fluid, and the blood. So a number of differences that may have influenced part of these results. Interestingly, if you look at the dose-response curve of the endotoxin assay, you can see that the curve has a specific characteristic, and after having surpassed the limit of 0.9, has a shape, as an asymptotic shape. In other words, after this point, it's very difficult to say how much is the concentration of endotoxin in the body, and can be several grams or some nanograms. This is absolutely difficult to say. But the most interesting thing is this window, which is something in between 0.6 and 0.9, where the endotoxin here is highly measurable. And indeed, it corresponds also to an interesting distribution in our population, where the vast majority of patients had indeed this window of dosage between 0.6 and 0.9. Above 9, the one where there was indeed some question marks and the difficulty in identifying the real amount of endotoxin is a minority. Interestingly, if you look at the capability of the cartridge to absorb the endotoxin, there is an average capability that is roughly 12 micrograms of endotoxin per each cartridge. But looking at the burden of endotoxin that can be calculated taking into account the endotoxin in an average size adult, you can see that as soon as you look at the high non-measurable asymptotic portion of the curve I alluded to, you may get an amount of endotoxin which is very, very high and may exceed the capability largely of the cartridge to absorb the endotoxin efficiently. If we look at the modified per protocol population, I understand that this is a post-hoc analysis, but it has several points of interest, and we are referring to a population where the endotoxin activity was exactly between 0.6 and 0.9, with a MOD score above 9, and who received the entire treatment. You at the end have 194 patients with the belonging to the modified per protocol population, 88 belonging to the PMX group, and 106 to the sham group. Again, analyzing the characteristics of these two populations, at the end you can see quite easily that they are quite balanced and resemble quite well to the original and larger population. And the same is true not only for the demographic characteristics, but also for the baseline variables in terms also of distribution of the various organisms. Well, as soon as you refer to this specific population, you see that there is a different result. And then you have a decrease of mortality rate at 28 days, which is quite significant, with a p-value of 0.0474, having a crude mortality that is 26% in the patient receiving the active treatment, almost 37 for patient in the sham group. And the absolute reduction in mortality is shown in the lower part of the slide, where at 14 days, 28 days, and 90 days, this significant remain constant. Interestingly, if you just look at this perspective from a different visual angle, analyzing also the time distribution in terms of hazard ratio, you can have a risk reduction that is 52% at 14 days, 42 at 28 days, and at 90 days, 41%. But one of the nicest things that almost at 3 days you can see the two curves that are nicely divergent. And analyzing and stratifying by the forest plot the mortality and including into this analysis the adjustment for through the page or the use of the norepinephrine dose or the reduction of the endotoxin activity, despite of the large confidence interval in some of these variables, in large part the odds ratio appear to be in favor of the polymyxin B filter. Interestingly, looking again to the secondary outcome per protocol population, you have some interesting results that in a way or another depicts also the improvement of the multiple organs score dysfunctions. Well, in the general population, even though the study was negative, you can have a greater increase in mean arterial pressure by 5 millimeters of mercury. And if you look at the dose of vasopressor, this is along the same line. And this reproduces also what happened in the study that we had in Italy, the other randomized trial published in the JAMA in 2009. Interestingly, we have the difference of ventilator-free days that was significant in this subgroup, as well as the renal replacement therapy 3 days. And in this case, the median difference over 28 days in these patients that were treated into the different groups. The subgroup analysis showed us another interesting point concerning the group of patients where no bacterial could be identified, but where in this category of patients the effect of polymyxin was indeed significantly better than in the sham group, as well as for those patients whose baseline dose of norepinephrine was above 0.1. So those patients were indeed more severe. Which are the conclusions? As first, the safety. The safety profile showed that there was no serious unanticipated adverse event, and very few of these events were related to the device, mostly the clotting, but obviously these were not a real serious adverse event. And the polymyxin is easy to use. In something like 95% of patients receive the treatment within the study timelines. And the 4% of the clotting was caused an early discontinuation. So the device safety metrics are certainly in line with the proven safe track that recorded of the PMX cartridge throughout over the 20 years in more than 150,000 patients. What about the efficacy? We cannot deny that the primary endpoint was missed, but this is, I think, one of the major findings of this study, there was a significant step forward because there was the possibility to demonstrate some benefit in a personalized approach. We, for the first time, understood a behavior of patients who had endotoxin, and probably has been identified the category that may have the best benefit receiving this kind of treatment. That is the category of patients whose endotoxin activity is indeed better measurable between 0.6 and 0.9. And the demodified per protocol population, exactly the range of activity I alluded to, demonstrated a 42% mortality risk reduction in 28 days. So probably we can also conclude that the subgroup analysis that I show you had a central role for the endotoxin and endotoxin removal, especially in patients with septic shock and not culture proven by bacterial infection, that we know quite well that at least in septic shock patients or those affected by any kind of infection may represent not less than 30% of all negative cultures. Despite that, these patients are in septic shock. So having also the possibility of offering an advantage to these further subcategory of patients may be of some importance. Thank you.